Welcome to a new episode of Idea Collider with some of my favorite people on the subject of innovation within the pharmaceutical industry. And I'm delighted to welcome someone I regard as the kind of J.D. Salinger, if you like, of the kind of pharma innovation space. Sort of pops up occasionally, writes this remarkable thing, and then sort of drops away quietly. And Jack's provided many of the hits in terms of thinking about R&D productivity and decision making within pharmaceuticals. So and Jack has maybe Jack, say hello to you. Maybe you could tell us a little bit more about where you've been and what you do now.
Yeah, well, thanks very much. So you mentioned periodic invisibility, and in fact that's partly related to what my professional life has been like for the last 15 years. So I have done a bunch of things, but I've spent some of my time in drug and biotech investment. And when you're in drug and biotech investment, it's a very heavily regulated business. So it's actually quite hard to have public opinions some of the time, or at least your public opinions only go to paying clients of the particular institution you're working for. But I've moved around between drug and biotech investment. I started out a very long time ago studying medicine. I think to the great benefit of health systems around the world, I never actually qualified, but did a PhD during my medical training that was in neuroscience. And I did a mixture of sort of wet neuroscience, i.e. real proper biological experiments, and computational stuff. I was then a university academic for a while doing the same kind of thing. I then went to work in the consulting industry where I sort of learned a bit about how the drug industry works. From there, it was into investment. And then I've sort of been back and forth between that, a little stint doing real drug discovery. I would say not enough to be a hands-on expert, but enough to know how hard it is, and some policy work. Most recently, I was back in investment. And as of the start of this year, I've been self-employed, effectively running a little consulting business.
Fantastic. And you first came across my radar with your original paper on the kind of better than the Beatles problem about R&D productivity, and that I think that spread very widely, very quickly about the sort of challenges of decision making. But you reflected on the challenges of R&D productivity within the industry, and how do you see that challenge? When you wrote it and since then.
So. I'll talk a bit about that 2012 paper first, because that may frame some of my other comments. But I was struck in the years running up to 2012, probably in the 5 years running up to 2012, how there was a sort of real mismatch between what appeared to be massively improving science and sort of optimistic technological rhetoric. So we'd sequenced the human genome. There's a whole bunch of new things we could do. Everything on the sort of scientific input side seemed to be getting hundreds, thousands, millions or billions of times cheaper. But I was working in drug and biotech investment at the time, and it was very obvious what the numbers were for the industry as a whole. And the numbers for the industry as a whole were bad and going in the wrong direction. So the industry was spending more and more, and R&D output was kind of bumping along at a pretty low level, despite exponential increases. And I use exponential in a technical sense, pretty much exponential increase in the amount of money being spent. So that by 2010, excuse me, you had roughly about 100th as many drugs being discovered per $1 billion spent on drug R&D as you did in 1950. And if you think about the quantity and quality of scientific knowledge in 2010 compared to 1950, that to me didn't make sense. And there was a lot of debate around R&D productivity being low, but I think not enough people had drawn attention to the contrast between improving input efficiency and improving output efficiency. And that seemed to me to make the problem more pressing perhaps than people thought. My view at the time was that the quality of the diagnosis of that problem in the public domain was low, right? So there were lots of very clever people in drug companies doing work on it, which we now know, some of which was successful, right? The regulators were doing some work on it, but it was not a major thing that people talked about. And that led me to be motivated to write the 2012 paper where I tried to identify and put names to, in some cases, things that people sort of knew were going on already. So we talked about the better than the Beatles problem, which I think is one of the main problems. And that said that, imagine how hard it would be to bring new music to market if every new record had to be better than the Beatles, if you could download all the old Beatles records for free, and if no one ever got bored of listening to the Beatles. Now, if you had those three conditions, it would be very hard to launch new music. Now, the pharmaceutical industry is unusual in that effectively you have those three conditions. So if we think about a drug like metformin, which is a diabetes drug, well, it's brilliant, right? It's like the Beatles in terms of its quality. Secondly, it is almost free because it's generic and people can make metformin. I don't know whether it's for 1 cent a pill or 5 cents a pill, but you can make metformin for next to nothing and it sells for next to nothing. And then, unlike records, doctors don't get bored of prescribing metformin. So one of the problems you have in the pharmaceutical industry is you have this ever-improving back catalogue of virtually free stuff against which any new medicine would have to compete. And that tends to push the areas where innovation would be economically valuable towards those areas where we don't have lots of great drugs. And however one defines easy or hard, if the last 70 years of the drug industry have failed to produce new drugs in a particular area, it's probably hard, right? So that's it's a very unusual problem the drug industry faces. And there are some other industries that face that problem, like, for example, crop protection chemicals. Arguably, they have that problem to other ones as well. But you've got this weird situation where you're an intellectual property business, but people don't get bored of the old intellectual property, and the old intellectual property is free. And I think that's a unique, it's an unusual challenge that very few industries face. And that, I think, is one reason why it's difficult for the drug industry over time to maintain its productivity levels.
And it's certainly the one that they like. The idea of, if you like, because it almost excuses one of the declines in productivity. But you go on to, attribute that decision-making, challenge. You've got that low-hanging fruit problem that, once that's gone, where do you go? Yeah. There is a big question about whether that's a finite pool or not, with, the number of diseases that we're addressing. And you talked at the time about, this kind of idea of reductionism within the mindset.
Yeah.
Could you elaborate on that for us?
Yeah. So, broadly speaking, right, if you've got a process where the inputs get hundreds, thousands, millions or billions of times cheaper, but the output gets 100 times more expensive over 50 years, there's two broad classes of explanation, right? One is we're running out of something, right? Now, arguably the better than the Beatles problem or low-hanging fruit arguments are that part, right? We've run out of something. The other part of the explanation is what we've done is we've effectively stopped doing activities that are genuinely productive, and we've substituted them for activities that are not productive. And I think it's now widely recognized. I mean, there's debate at the margins, but I think it's now widely recognized that the first wave of what you might call industrialized target-based drug discovery, or I'd call it target-based drug discovery version 1.0, was not a success, right? So I think there was an idea in the industry that grew up probably in the 1980s, partly, I think, because people realized that the productivity of some of the older methods was declining. There was a sense that you could sort of industrialize drug discovery and you could build these big factories. And if we used genomics or other methods to identify drug targets, and then if we screened very, very large compound collections against those drug targets, that would inevitably, we could sort of build an industrial process that would just mean we could very quickly and very efficiently discover cures to lots and lots of drugs. And for various reasons, that at least version 1.0, I think most people now recognize was a failure. Now, that sort of relates to, I think, what is a sort of wider debate in sort of biomedical science and biology in general, for which people use the term reductionism, right? And this is the idea that somehow, looking at individual molecules, maybe the sort of best or most, the truest way of looking at biological systems. And I think I had an intellectual prejudice against that, which may be false, by the way, But I had an intellectual prejudice against that, partly because of my own scientific training. So I was a neuroscientist. And arguably neuroscience, if you had to pick one biological discipline where reductionism is problematic, neuroscience would be that discipline. So, I went through my PhD and subsequent academic career knowing that, you couldn't predict how proteins fold from knowing their amino acids, right? You couldn't predict protein function from knowing how proteins fold. You couldn't predict nerve cells very well from knowing the channels that were in their membranes. You couldn't predict the behavior of neural circuits from the behavior of neurons. You couldn't predict, the behavior of bits of brain from the neural circuits in them, right? So that being my sort of academic background, having lived and looked at the drug industry from sort of 1996, 97 to about 2010, we were meant to believe that sort of doing genomics, right, identifying these individual components of biological machinery was somehow going to be very predictive of clinical utility in man. Now it turns out it sometimes is, right? But I think there was a sort of naive optimism that sort of circa 1985 to sort of 2000 that kind of molecular reductionism was the way to go. And I think things have changed since then. But I certainly think some of the decline in productivity was self-inflicted because people made the wrong technology choices.
And we've kind of industrialized that decision-making process as well, so that, there's kind of, as you say, there's a belief in the glimmers that we're getting that this is enough to become predictive, rather than embracing the idea that it's hard to predict from some of this stuff. So I think there's almost undue faith being placed in some of those new technologies to do the whole job for us.
I'm going to agree with you, and also I'm going to agree with some of the things you've suggested in some of your recent sort of productivity indices, right? So in 2015, I wrote a paper which was called something like clinical selection versus intelligent design, right? So there's two kind of archetypical models, or there's two extreme ways one could imagine new drugs are created, right? One way is you do some genomics, you find a gene and its associated protein that are associated with a disease. You then find a compound that targets that protein. And at that point, or that's kind of all you need to do to predict what's going to happen in man. Right. So effectively, you have a kind of intelligent design process where from the sort of genomic insight and the medicinal chemistry, you effectively have ready-made a therapeutic that is good for man, right? The other extreme would be a model where you say, actually, all the drug industry does is produce plausible chemical diversity, and that's a term that a former colleague of mine invented, which I think is a good one. So the drug industry makes plausible chemical diversity that you then throw into the clinic, and the clinic, doctors, clinical trials, patients, then select what works and what doesn't. Right. And you can imagine a whole bunch of different sort of intermediates where the useful knowledge is gained at different places. And my strong suspicion is that actually much of the innovative heavy lifting actually happens later in the process than many people like to admit. And that we tend to systematically overestimate the importance of intelligent design, important as it is, largely because of survivor bias. Right. So every drug that comes to market with a lovely story that explains how it works. Now, quite often we subsequently find that those stories aren't true. Right. And we change the story. But because every drug at every step of the process until it fails has a nice rational story that explains how it works based on its receptor binding, et cetera, that fools the world into thinking that those scientific stories are more predictive than they actually are. And I also think that's reinforced by the way intellectual property rights work. So at the moment, it just so happens that we're in a world where novel chemistry against a target is particularly appropriable, right? That's what you can make money from doing. So because of the survivor bias, we hear stories that suggest drug R&D works like that. And because novel chemistry is particularly appropriable, that's where the money goes. And my view is we actually miss quite a big trick, because much of the innovative heavy lifting happens in other parts of the R&D process.
or happens serendipitously,
or happens
something
serendipitously.
Like yeah, and I think, David Epstein talks about this in range, that idea of premature specialization, across many fields being part of the problem, that, as you say, stuff gets put on the stockpile or the shelf once it's failed, but it failed at something instead of failing, full stop. And
sorry, could I say, and also I think it's quite striking how history gets rewritten, right? So if you look at a very successful drug called Avastin, for example, Avastin was invented with the idea that it would starve the blood supply to new tumours, right? I think there's at least a plausible working hypothesis now on how it works, which is it normalises tumour vasculature, which is almost the opposite thing, right? Another example would be Gleevec, which when it was first discovered was originally sold as a highly selective inhibitor of a particular fusion protein, right? Well, actually now it's used in a bunch of other diseases, and we know it does a bunch of other things. People call it a broad spectrum agent. So effectively, the history of drugs gets rewritten to match the kind of creation story.
And which comes first? I think that I remember working on Avastin, before launch, and choosing the stories about, was it chemosensitizing, was it normalizing, was it starving, but you pick a story and you run with it, and then and then people start believing it, and that becomes. But I think it loops back to the kind of conversation about the Beatles, because remember the Beatles were an odd bet at the time for a company, and their early days were spent as a covers band, so they were hard thing to predict what they would become, and actually it was the development process that kind of honed the Beatles as opposed to what they eventually became. So looping back to this productivity issue, you've drawn an analogy with the oil industry, which I thought was interesting. Could you mention that?
So, and this is also partly to do with the sort of debate on what it is that's driven productivity in the drug industry, right? So productivity arguably has improved a bit since 2010. But for me, the really interesting thing is this massive, 2 order of magnitude decline in number of drugs discovered per $1 billion spent, despite massive gains in all of the tools that people who do drug discovery think are important, right? And imagine that you had something similar in the oil industry, right? Imagine that BP or Exxon or ENI were finding it 100 times more expensive to suck a barrel of oil out of the ground now than they did in 1950, but their drilling technology got much better, right? The debate you would have under those circumstances wouldn't be about lots of things that people think are important in the debate around R&D productivity. So people talk a lot about kind of organizational structure. Should you be in-licensing more or doing your own drug discovery, right? What should the organizational structure be like? What should the reporting lines be like? How do you design incentives for people working in R&D? Now, I'm not saying those things aren't important, right? But to me, they don't seem to explain the scale of the productivity changes that the industry has seen. So that it seems to me but the most serious discussions and debates around R&D productivity actually need to look at the kind of factors that can drive orders of magnitude of productivity change. Because if they can't drive orders of magnitude of productivity change, they're probably not really the thing that has meant the industry has ended up where it is.
Yeah, and that idea of, the models that are used for screening, the models that are used for disease, kind of, as you've written, play into that significantly. So I'd be interested in, you telling us more about those models, and also linking that to, I'm going to say, the kind of managerialism, the kind of McKinseyization, if you like, of the decision-making process, the incentivization of discovery and development. I like that idea. I think you've written that, if you put 5 different people in a room, depending on where they come from, they'll make different decisions on the same molecules. So, I'd be interested in your thoughts on screening and disease models at that very early stage.
Okay, I'll talk about the model stuff first, and then the sort of managerial side. And if I forget to talk about the managerial side, because I sometimes get carried away on the model side, remind me, okay?
Yep.
So after the sort of 2012 paper that talked about things like the Best and the Beatles problem, and other things that are important, like tougher regulation is important, possibly sort of over-reliance on reductionism is important, I I think I sort of thought for a while on this, or I ended up thinking about R&D as a formal search process, and this is going to sound a bit arcane for a while, but I'll try and sort of bring it back to reality. I remembered from my dim and distant academic past that certain sorts of search tasks are the efficiency with which you can perform certain kinds of search tasks is very sensitive to the precise nature of the search algorithm you use. So not all search algorithms are equal, and different search tasks you'll do better or worse with different kinds of search algorithms, and those effects can be quite powerful. And it struck me that actually drug R&D is really a kind of search process where you have lots and lots of things that might be useful. These could be drug targets. They could be the compounds in a screening collection and the analogues that one could make from those compounds. They could be the monoclonal antibodies that one could reasonably make against a drug target, right? So in general, in the R&D process, what you're doing is you've got lots and lots of things that might work, but which individually are very unlikely to work. And then you have a set of measures that you apply to those things to try and identify useful. And this changes as you go through the R&D process, right? So at the start, you typically use quite cheap measures that you can apply to a lot of those things. High throughput screening would be an example. And later in the process, you have very expensive measures, such as a phase 2 trial or a phase 3 trial, that you can only apply to a very few of those things. But throughout the process, you're using those measuring devices because you think their output somehow correlates with clinical utility in man, right? So you may not think high throughput screening correlates very highly with clinical utility in man, but if you didn't think it correlated at all, you wouldn't use it. There'd be no point using it. So you can sort of imagine a kind of point cloud, right? So you've got all of these therapeutic candidates that may or may not be useful, and you've got a bunch of measures that you can apply to them, ranging from high throughput screens, toxicity screens, various computational measures around solubility through to, animal models and then early human trials. And you can imagine representing each one of those therapeutic candidates as a kind of point in some, multi-dimensional graph where its position would be specified by where it sat on all those measures. And this is very arcane so far. So why might that be a useful way of thinking about it? Well, if you do that, you can then start trading off different parameters in your search process. So you can say, well, okay, how, for example, would I trade off validity against throughput? And validity you can operationalize. You think, well, validity is the correlation that would exist if I tested lots and lots of items in my model and then if I tested them all in man. Right? That's the validity of the model, the correlation coefficient. Throughput is, well, okay, if each individual assay is expensive, maybe I can only test 10 items in that assay, in which case I would expect an average to be able to find the one item in the top 10%. If I could afford to test 100 items, I could find one item in the top 1%. If I could afford to test 10,000, I could, find the best 1 out of 10 to the 5. So you can make a little quantitative model where you can trade off throughput against the degree to which your model correlates with the human outcome of interest. And if you do that, the results are genuinely surprising, or they surprise me. And I suspect they should surprise people in the drug industry as well, because it turns out that when positives are rare, i.e. if you're searching and only 10% or less of items would, if randomly selected, work in man, and good drugs are rare, right? We know that most randomly selected chemicals won't be useful for a particular human disease. It turns out the very small changes in the correlation coefficient between your model and the human outcome of interest are often more important than 10 or sometimes even 100-fold changes in the number of items you test. Right. So this perhaps starts to provide a slightly better explanation for the decline in R&D productivity. So imagine we've got a world in which you've got a bunch of different screening and disease models circa 1950, right? And some of those screening and disease models are really good, and some of them are totally unpredictive of whether a drug or not will work in man. And in 1950, medicinal chemists are pretty clever by 1950. They can make lots and lots of useful analogues of compounds. So you start using these models. What you'll find is that the models that genuinely predict clinical utility in man give us drugs that work. And that means over time those models become commercially and academically redundant. right? And it turns out that anti-infective models are pretty predictive. We've got lots of really good antibiotics. Drugs for stomach acid secretion are pretty predictive. We've got lots of really good stomach acid drugs. So the better than the Beatles problem, in my view, is somewhat explained by those areas where we have good screening and disease models. So there's a very high correlation between we've got lots of good drugs because we've had lots of good screening and disease models for a long time. So the best models become commercially and academically redundant. They get retired. What's left? We're left with the screening and disease models that don't work. And ironically, because they don't work, the diseases don't get cured, so we keep using them, right? So the models that are most likely to be used for the longest, ironically, are the worst models, like, for example, rodent models of Alzheimer's disease and lots of models of solid tumours. We keep using them precisely because they don't work. So, coming back to your managerial question, unless you run the decision theoretic maths, you wouldn't have realised that, right? You wouldn't have realised that tiny changes in model validity are more important than doing 10 or 100 or 10 or 100 times more experiments or spending 10 or 100 times more money. My guess is, because that's a non-obvious result, is that managerial common sense very often pushed the drug industry the wrong way, right? So if you don't know how important model validity is, it's not an important enough parameter in the way you organize companies, in the way you make decisions. So it's very easy to ask people, how many analogues did you synthesize today, or how many compounds did you screen? It's very hard to say how valid was the model, because that's based on sort of expert scientific judgment. We never know for sure until it's too late, right? Very, very hard to manage. And I think because it's hard to manage and people didn't realize how important it is, it's been undermanaged.
And it's really interesting, that idea of validity, model validity, and maybe even, multiple models and triangulation between them. It's not a subject that I hear discussed, and I think you introduced that idea that even the kind of priors or the beliefs of the people, making the decisions with the models in front of them can, that's a model in itself.
Exactly. So I'll give you a concrete example, right? Or rather, the maths is quite generalizable, right? So when I explain it to people, I think they typically think in terms of, when I talk about a model, I must mean a particular high throughput screen or a particular, rat with diabetes, right? Well, actually, the logic applies to anything during the R&D process that is ranking candidates and then making decisions about them and putting in a threshold. So a model would be the gut feel of a senior medicinal chemist who looks at a bunch of compounds and goes, oh, I don't like those structures, right? That logically would serve the function of a model. And similarly, the briefing documents to a portfolio management committee would be a model. And I'm going to give you a sort of anecdote here that is based on my own experience. But I, a little while ago, had to do some reviewing for one of the UK biomedical funding agencies that was looking at university grant applications for anti-infectives. And this was before I did this work, by the way. And arguably, the single most important thing the committee should have known before allocating money to particular anti-infective discovery projects was the degree to which one could believe the outputs of the models as being validly predictive of what would happen in a human, right? Now, there was almost no argumentation about that at all. So each grant would tell us how much money they needed, over what time period they were going to spend it, how many FTEs they wanted, what they expected to publish, right? What things the PI had published before. But the single most important thing that people on the committee should have been told about, we really weren't told about. And then secondly, because we on the committee didn't know that was the single most important thing, we didn't ask for it. And then even if we'd been given it, we probably wouldn't have understood it because actually, if you'd realised that model validity was the most important thing to opine on, you'd have put different people on the committee, right? Now, I suspect many drug companies are not in quite that situation, but I do suspect that unless you really think about validity and sort of try and train people on it and develop sort of institutional norms around it, you'll end up making the wrong decisions. So, for example, if you've got a psychopharmacologist, a cancer biologist, an infectious disease specialist looking at the same documents from therapy areas in which they're not absolutely expert, they will have their own views about whether or not to believe the rat model. which will be different depending on which of those three disciplines they're from, right? So I think a sort of people should take a more kind of validity-centric view. It's not easy to do. A lot's done already, but I do suspect there is more that could be done.
Yeah, and you've written about or thought about that kind of idea, why you end up with more cancer drugs at the moment than other things. Because probably we have a relatively rich environment in that world, where some of the models in cancer are slightly more predictive of other disease areas.
So I think there's been some improvement in cancer. I think the cancer uptick is really to do with, and rare disease uptick is to do with three things. I think it's to do with money, right? So R&D capital follows profits, right? So where people, and pricing is, the pricing environment is very benign in oncology and rare diseases. I think in oncology, the molecular slicing and dicing of cancer subtypes means that you can now match in vitro and in vivo models better onto human population. So essentially, what you do is you effectively, the model becomes more valid because you can now apply it to the patient population to whom the results of the model apply, right? So you see that particularly with, sort of the targeted therapies. I mean, not so much with immuno-oncology, but with targeted therapies, yes. And I think that's one of the reasons we've seen an uptick in cancer drug approvals.
Yeah, and I think there's an interesting thing there, which is, because I think cancer itself has changed as a diagnosis, right? It's become more, closely defined. If you take something like schizophrenia, you don't have that same, luxury. You've got largely, old disease categories that are, broadly applied to very specific, compounds. I think, Derek Lowe, was, who wrote about your paper a while back, he's long argued about the D1 hypothesis in schizophrenia as one of those models that doesn't make sense. So I guess what we have in pharma is this kind of very odd feedback loop, very long cycles. So a lot of the folks within pharma don't have knowledge of what happens with the models that they're applying. So if we move into the sort of ideas of practicality, or what could be done practically with the knowledge that you've uncovered of model validity, what could we be doing differently?
Okay, so I'm going to start with model choices. There's some other things. So again, if we've got time, remind me, and I may talk about some of the other things. But I think the model choice point is a very interesting one. And use the schizophrenia example. I don't know much about schizophrenia, but I'm going to use the example of depression because I've talked to some people. Again, I don't pretend to know about it, but I've talked to some people recently who do know about it. And one very experienced sort of translational researcher in depression I know said something to me quite striking, which I have no reason to disbelieve, and that is that not a single antidepressant drug class was ever primarily discovered with an animal model. So his argument was every single antidepressant drug class we know was discovered in humans. Now, you may subsequently have been able to make an animal model that would predict other members of the class, but every novel class came from an observation in man. The only one that I think people would argue about would be the SSRIs. Right, but again, his view was that actually that was discovered in man, despite the fact there was pre-existing animal data. Now, so how does one address the problem then of novel antidepressants? Well, in 2010, or around 2010, maybe a year or two before then, there was a very interesting observation made, which is that antidepressant drugs are peculiar in that they cross the blood-brain barrier and start doing things in your brain relatively quickly. But if someone is diagnosed with depression, you give them an antidepressant. It typically takes them between 4 to 6 weeks before they notice before there's measurable mood changes. Now, in around 2010, maybe a couple of years before that, it was noticed that if you get people with depression and you get them to rate social cues, i.e. you show them faces and you say, is this a happy face, a sad face, they tend to have a negative bias in their perception of faces, right? Or social cues. If you give them an antidepressant with one dose, you can you can tend to normalize their perception of social cues. So this leads to a hypothesis of depression that actually what you have is you've got some error in perception of social cues, for example, and that then drags your mood in a particular direction. But also, if it turns out that you can look at short-term changes in perception of social cues as a genuine predictor of antidepressant potency, then actually maybe you've got a kind of translational model of for testing novel antidepressants, right? So yeah. This is a sort of a kind of experimental medicine type approach. I'll give you another analogy, and this is not necessarily a real example, but it illustrates the thinking. Imagine that one wanted to find a drug for a neurodegenerative disease, right? Well, we already know that Alzheimer's and Parkinson's are really hard and that the rodent models are pretty poor. Well, maybe you choose a different problem. You choose the problem of chemotherapy-induced peripheral neuropathy, which is the dose-limiting toxicity of drugs like carboplatin and cisplatin. Now, my suspicion is that you could actually get tissue which would show what the peripheral neurons of people who are suffering from chemotherapy-induced peripheral neuropathy look like, and that's because, unfortunately, cancer patients often have to have surgery, right? One could characterize those neurons. One could give a rat carboplatin or cisplatin, and one could characterize its neurons, and also you could look at its behavior to see if it seemed to have the behavioral symptoms of neuropathy, right? And then one might be able to, for example, take neurons out of the rat and grow them in culture and see if you gave them carboplatin or cisplatin, they actually resemble the neurons in the rat. Did the electrophysiological properties of the neurons change in the same way? So what you do is you start with the human pathology, and then in teeny tiny baby steps, you work back, satisfying yourself at each point that what you've got is a valid model of the step above. And you do that only as far, the minimum number of steps necessary to have something in which you can screen drugs in, cognizant of the decision theoretic result, which is if you've got a good model, you're much better off screening a few hundred things in it than you are screening 10 to the 7 compounds in a lousy model, right? And I think some parts of the drug industry and academia have kind of got that idea. I think they got there a different way. So if I look, for example, at Vertex, I think the way they think about the problems that they choose to tackle is again based on this idea that we only choose to tackle problems that we think we can model, even if that introduces, for example, risk in terms of therapeutic modality we need to bring to bear. I was reading some material from an outfit called the Structural Genomics Consortium in Oxford as well, who are trying to produce disease models. And again, I think their philosophy for producing disease models is or human tissue-based resources is kind of similar, right? You try and take these baby steps back from the disease rather than starting off with some sort of simplified molecular representation that you hope is going to predict what happens in man.
Yeah, because I think, you're exactly right that we've got an industry that's largely modeled itself on enough proof to move through the stages to the same steps that everyone else is going to do. So you prove validity to go to phase 1, you prove validity to go to phase 2, but it isn't necessarily a disease validity. It's there's something else. It's part of a nutritional based model, which is, if we just do enough of it, something will come out the other end, and that will be valuable enough to pay the bills, which is starting to become a problem within the industry. So, in terms of that practicality, I think some of that early stage validity, you've kind of, as you described it there, or the models or the creation of this stuff, that doesn't have to be done by a pharmaceutical company, right? I mean, there are lots of entities that could be doing this very early stage stuff that, without that problem.
Yeah, I think that's right. And in some ways, I think at the moment, there is a sort of economic problem, which I've only really started thinking about. I think other people have been thinking about it longer than me. But there is a issue at the moment, I think, which is to do with what economists call appropriability, right? And I think I might have mentioned this a few minutes ago, that it turns out the thing that's easiest to secure robust intellectual property on for the drug industry is novel chemistry, right? And it's quite hard to to capture the value from it's quite hard for a private sector company to capture the value of innovation and screening of disease models. It's not impossible, but it's difficult. And the reason for that is, let's suppose that, you're a drug company working in Alzheimer's and you said, right, we're really going to throw everything at understanding the disease mechanism, and we're going to be incredibly creative and innovative and spend a huge amount of money coming up with a rodent model that really predicts what happens in man. Well, even if you could do that, the minute your drug hit phase 2 and showed a positive result, every other company that hadn't made that investment in the disease model would know that the mechanism you'd been working on was valid, right? So you would effectively lose the value of that invention to your competitors, even if you patented the model, right? And I think what that means is, and you see that, I'd say, oncology in some ways has a lot of drugs being developed at the moment, but boy, oh boy, there's a lot of drugs being trialed. And if you look at oncology at the moment, you've got this really weird situation where you've got lots of companies able to raise money or spend money doing novel chemistry, and they're all spending it on disease models, often outsourced, that they all believe are rubbish. Right? That is not a sort of optimal solution for society, right? But it's because the chemical novelty is appropriable, the disease model novelty isn't. People underinvest in disease models. So my suspicion, at least in the near to medium term, is that a lot of benefit could come from public sector biomedical research funding agencies and some of the biomedical charities actually trying to correct that bit of market failure and doing some of the disease model development and calibration that it's quite difficult for private sector drug companies to invest in.
Yeah, it's very interesting. I think that, I know that I've written about the idea of, the competitive advantage for companies is going to be about their ability to learn rather than their ability to just do, or apply the same process as everyone else. And I think that is absolutely part of that challenge, which is that we've always created a doing industry rather than a learning industry, despite the, as you said, this kind of, serendipitous, discovery along the way, often producing our successes instead of the failures. And then you layer in all of the other things that you need to know about, market performance, you need to know about formulation device, the diagnostic, all of those other things. So not just about the molecule model. One thing I'm going to do is make sure that in the kind of show notes, people get links to your paper so they can dive literally as deep as they want to into the decision making sort of algorithms. Maybe you want to loop back to your thoughts on innovation in general. What do you define it in a particular way?
Well, by the way, I've watched some of your previous interviews, so I guessed you were going to ask me, and I actually realised that I didn't have a particularly good off-the-shelf answer. So I scratched my head. And then, to be honest, I think the general principles of patentability. Now, I'm not going to say the way those principles are implemented, but the general principles of patentability, I think, are not bad shorthand for thinking about innovation. And effectively, in the US, stuff that can be patented has to be novel, has to be new, has to be not surprising, and it has to be useful. Now, I actually think that's not a bad summary. So innovations are things that are novel, they're not obvious, somehow surprising and useful. And I think in general, the importance of innovation or its impressiveness depends on, some aggregate in score across those three dimensions. But I think I would say a couple of things though. One is, I've been very impressed by a book I've just read by a guy called Robert Gordon, and it's called The Rise and Fall of American Growth. And that makes a wonderful point that there are certain phases of human history and certain geographic regions where innovation seems to be remarkably rapid and relatively easy. And then there are huge swathes of geography and time when not much happens at all. And I'm misquoting the book, but there's a wonderful line in the book where I think he is misquoting someone else, where he says, if you think about sort of economics and growth, right, and GDP growth follows from innovation, right, the argument is between the fall of the Roman Empire and 1750, for the vast bulk of people who lived on the planet, nothing happened, right? I.e., for the lives of the average person between the fall of the Roman Empire and 1750, there was no innovation. Right? but between 1870 and 1970 in the US, you had the creation of the modern world, right? So by 1970, the average American kitchen would have in it every single kitchen appliance that we have today except a microwave oven. Right? That's the only thing. Mm-hmm. By 1920, pretty much every American household that wanted one had a car. In 1900, virtually no one had a car. Mm-hmm. So I think it's very easy for us to sort of look at particular, So I think what I'm saying is if you live in the Bay Area, between 1985 and 1995, innovation seems relatively easy and normal, right? But I think it's not, right? So most of human history and most of the time, you don't get a huge amount of innovation. So it's peculiar, and a whole bunch of things have to come together, right? It's almost like not the norm. It's not the natural state of man. And I haven't really thought about what those things that have to come together are, but that strikes me as quite an important point for anyone who runs a company or is practically involved in sort of policy and so forth. And then I think the other thing, which we sort of alluded to a bit, is the intellectual property and sort of the incentives really matter. So at the moment, society, I think almost, not by accident, or rather, patent law at the moment means that we get a lot of certain kinds of innovation, and you don't get financial incentives for other kinds. So as you said, right, creation of novel chemical matter seems to have a privileged place at the moment. And there's a whole bunch of other stuff that is very, very useful, but we perhaps don't incentivize it enough.
No, it's actually one of the interesting challenges. And do you think that we're at a point when, if you think back about decision quality and model validity and so forth, that things like AI, that other technologies are going to make a dent in the in our productivity and our success rates?
So I don't want to sound more negative on AI than I am, right? So one of my brief stint actually doing drug discovery was in a little company that at the time it said it was doing network pharmacology and bioinformatics, but if it was raising capital today, it would definitely call itself an AI company, right? I think AI will be very, very useful in certain constrained niches, but I think it will make less of a difference on the drug industry than it's made on most industries. And there's 2 or 3 reasons for that. The first reason is the drug industry has been doing what other industries call AI for quite a long time. And for my previous employer, when I worked at the bank, we wrote a massive report on AI, or rather, some of my colleagues wrote a massive report on AI, which I had to contribute a small section. And I remember going back and just looking through the literature, and I thought circa 1985, you could find papers where people in the drug industry were doing and publishing stuff that, if it were done today, would be called AI. The drug industry is unusual. You had lots of statisticians, computational chemists, geneticists, right? They've been in the drug industry for a long time, and they've had a lot of money and big computers to play with stuff, right? That's not true in retail, right? So Netflix and Amazon are doing clever stuff with AI, not because the statistical algorithms are particularly new, but because they've got lots and lots of data that they can capture in real time and then analyze in real time and put back into the hands of consumers in real time, right? So, so I think, the drug industry, you've got this thing where you've got this history of kind of aggressive quantitative data analysis. I think, again, you say weather forecasting may be a similar kind of thing, right? No one says AI is going to revolutionize weather forecasting because weather forecasters have had statisticians, physicists, and supercomputers for quite a long time. And then I think there's 2 other things that constrain things in the drug industry. One is, at least in biology, there aren't good predictive theories, right? So, and then also in biology, or rather, you've got data constraints as well, and not entirely, but roughly, the incumbents are the people with the best data, right? So, and it would be very, very, very expensive to generate a lot of the data. Now, I don't want to make the incumbents' data sound better than it is, right? So people who work in big drug companies will pull their hair out at a lot of the data problems they have. But broadly speaking, they've got, a lot of them have a lot of data. So I think in particular niches, like certain medicinal chemistry niches, and other places, it will be very useful, and it is already very useful. But I don't think it's going to. I wouldn't expect a step change in drug R&D as a result of AI. Now, there's a bit of your question which I've forgotten what it was. In giving you that long and tedious answer.
No, I think you did address it, because I think that challenge of prediction almost needs that feedback loop, right? And we have the additional problem of weather forecast type prediction tells you broadly whether something might happen or might not happen, and we kind of use that because it's got a rapid feedback loop. Of course, we don't have that luxury within our space. So I think it's I guess the question is whether we have these adjacent technologies making a difference.
But I do think in some areas there could be a lot more model testing or back testing. So, for example, in oncology, there's an awful lot of drugs now that have gone into phase 1 trials, right? And there are sort of sporadic, piecemeal attempts to calibrate models, right? So if someone, for example, someone has a new organoid technology, there may be some attempts for a limited set of compounds to show that, the organoid is more or less predictive than a cell-based model. But I don't see a sort of industrial strength kind of calibration where people systematically go across very, very large numbers of compounds and different sort of screening and disease model technologies and try and look at all of them in all of them, right? And in those cases, even in the limited cases where it has been done, for example, where people have looked across cancer cell lines and looked at the same drugs tested in the same cell lines in different labs, there what you find is sometimes rather non-congruent results, right? So you're right that it's not like weather forecasting, and validation is always going to be a sort of very, very imprecise science in the drug industry, and by the time a model is truly validated, it's redundant, right? But nonetheless, I think in some therapy areas where lots of drugs have gone into man, an awful lot more could be done, but at the moment, the incentives aren't there for people to do that calibration and validation.
Yeah, it's almost easier to start with fresh material rather than to revisit. I'm aware that I've used up a lot of time already. Maybe a couple of things to kind of leave on. One is, taking this forward for the next 5 years. Do you see changes, or are you optimistic that this kind of complexity, decision quality, thinking, validity modeling, all of these things will make a difference to the industry, or pessimistic?
I would say I am. I think there is at the moment. I think there is probably one dominant idea about improving validity. Maybe I'm being slightly unfair. No, I think there's one. There's a lot of other ideas, but there's one dominant idea which I think will help. But the trouble is, I think too many people have got the same idea. So, and the big idea is human genetic validation. So there is some evidence that suggests that certain sorts of human genetic data can improve the odds in drug R&D. And it's most obvious where you have what's called sort of strong Mendelian data. This is, for example, where you have got one family who show some rare phenotype. For example, they have very tough bones, right? And that may help you discover an osteoporosis drug. And then there's another sort of evidence that comes from what's called GWAS, where typically you're looking at genes that have small effects on common diseases at the population level. Now, GWAS, I think, hasn't yet proven as useful. But nonetheless, I think I'm actually somewhat sympathetic to the logic that GWAS may become more useful. But the problem is, and this is a problem for investors, it's not a problem for society, right? The problem for investors is a lot of people have access to similar kinds of genomic and genetic information, right? Which I fear may mean that from an investor perspective, what you find is that you find a lot of competitive crowding in those areas where the genetic data is strongest, right? So if you've got the combination of strong genetic data in a benign reimbursement environment, that's where the drug industry is going to throw its money. And I think what that may mean is that you have a lot of competitive crowding. And I think, so for me, the interesting thing is and if I was back in investment, this is what I'd be looking for. Could one find therapy areas where there was ways of improving models or finding novel models, like that's a depression example I gave you, which are not dependent on human genetic validation, because those may be areas where actually productivity can go up, and you don't have a million other companies trying to deploy lots of capital in the same area.
Interesting. A kind of 90-degree idea instead of straight ahead, which, my last interview was Matt Ridley, and reading his book, no one would have backed the Wright brothers at the time, right, and everyone was trying to, figure out how to fly a plane because they were the non-obvious, people to do that. But, in hindsight, they had a good reason to come first with what they're doing. This has been wonderful, and, that I could ask you questions for another hour. I'll sign off. One thing, you made a reference to a book that you found particularly interesting. Are there others that you would recommend we go look at?
Coronavirus has given me a bit more time to read than I normally have. And so I'm going to mention two more books. I mean, I'll remind you the first one. The first one's called The Rise and Fall of American Growth. It's by a guy called Robert Gordon. I think more than any other book I've read recently, that explains to me how we've ended up with the world we've got, right? Really interesting read. There's another book called with Rise and Fall in the title. It's a great book, I think, for anyone interested in the biopharmaceutical industry and medical innovation. It's called The Rise and Fall of Modern Medicine by James Fanu. It's written by a different guy at a different time using different data, but in some ways it's remarkably reminiscent of the Robert Gordon book. So The Rise and Fall of Modern Medicine by James Fanu. And then there's another book which I've read which has got nothing to do with work at all, but it's just really unusual and peculiar, and left an impression on me unlike almost any other book I've read before. And it's a book called The Tartar Steppe by a guy called Dino Buzzati, The Tartar Steppe. And different books, fiction typically evokes different emotions in you. You can read a thriller to be thrilled, or something frightening to be frightened, or something nice to be buoyed up by. This book makes you feel really uneasy. Now, people may not want to feel uneasy, but it's a book about a guy who sort of effectively wastes his life. And you can sort of see it coming through the book, and he can't. And you sort of read it feeling uneasy and hoping he'll make different decisions, but knowing he won't. And but in terms of an impression, it really made an impression on me, and I can still sort of have that nasty feeling in my stomach every time I pick it up. So if you're feeling too jolly and happy, I suggest you read Dino Buzzati's book, The Tartar Steppe.
Fantastic. Excellent. Well, I think I will pick that up, because that sounds like this is a time of uneasiness in general, right?
Or if everything's going too well and you want to sort of,
Yeah. Fantastic. Jack, thank you so much for this. It's been a pleasure to reconnect and to hear more what you're thinking. So I'll post some links to your papers on the show notes, and if people want to find you more easily, what's the best way?
I will maybe put a link so you can have my email address or something. That's probably the best thing, but I'll send you that as well.
Fantastic. Okay. Thank you so much, Jack.
Okay. Thanks very much. Bye.
Thank you.